Hello students, myself Om Prakash Katte and you are welcome to Biology Reward. Today we are discussing class 12th biology chapter molecular basis of inheritance, and chapter topic we are discussing, it is structure of DNA. Structure of DNA. In the previous chapter we have learned that how is polynucleotide chain is formed. So let's now we have to discuss about the DNA. DNA stands for deoxyribonucleic acid, and this DNA is polymer of deoxyribonucleotides. DNA is polymer of deoxyribonucleotide. When many deoxyribonucleotides are formed together, they form the one long chain, and this long chain is called deoxyribonucleotide chain. So in earlier we have learned what is nucleotide. In nucleotide we have learned pentose sugar, this type pentose sugar, nitrogenous base attached here on the fifth position. It is the molecule of phosphorus, phosphate molecule. So it is one nucleotide, and here sugar is deoxyribo sugar. So this is the one molecule. Another molecule is this one, pentose sugar. It is another nitrogenous base, and it is the phosphate group. Then this, like many nucleotides units are combined and it forms the long chain. So DNA is long polymer of deoxyribonucleotide, because it is the ribonucleotide, this is nucleotide, and here is a deoxyribo sugar, so it is called deoxyribonucleotide. This DNA, such type of stand of polynucleotide will form, and in this polynucleotide there is the deoxyribo sugar, so it is called deoxyribonucleotide. So DNA is long polymer of deoxyribonucleotide. Second, DNA length, it depends on number of nucleotide. So what is the length of the nucleotide? What is the length? DNA length, it depends on number of nucleotide. So how many nucleotides are present? That determines the length of the DNA. If there are more nucleotide, the length will be more, and if there are less nucleotide, the length will be less. Look at one example. It is bacteriophage. Theta into 174 is bacteriophage. There are 5,386 nucleotide. Lambda bacteria, here 4850 nucleotide are base pair. E. coli 4.6 into 10 to the power 6 base pair, it means nucleotide. BP stands for base pair, or we are using nucleotide. Similarly, haploid content of human, haploid content, it means in ova as well as in the sperm. So in haploid content of the human, so number of nucleotide are 3.3 into 10 to the power 9 base pair, it means nucleotide. So in haploid content it is present. In defined content in the human, 6.6 into 10 to the power 9. So these are the number of nucleotide in DNA of different organisms. DNA is acidic substance, and this DNA is identified by Frederick Nisser. Frederick Nisser Patrick Research identified the DNA, and this DNA he identified in the nucleus, and he gave the term as a nuclein. So DNA is acidic nature, identified by Patrick Research, and in 1869, and he gave the term, it is nuclein word is used because it was acidic in nature and found in the nucleus. Second, James Watson and Francis Crick, these two scientists, they studied the detail about the double helix structure of the DNA. Many times question is asked, who proposed the double helical structure of DNA? So James Watson and Francis Crick, they proposed the double helicals model, and they were awarded by the Nobel Prize also. They studies by X-ray diffraction method. Okay, so X-ray diffraction method, they have studied the structure of DNA, and on this basis they said that the DNA is a double helix structure. We have discussed here. So James Watson and Francis Crick proposed the double helical DNA structure. Then Chargaff's rules. So Chargaff studied the composition of the nitrogenous bases in the DNA, and it is called Chargaff's rules, or it is also called equivalence rules. According to the Chargaff's rules, adenine always equal to thymine, or ratio of adenine and thymine always 1. This is the first rule. Second, cytosine always equal to guanine, or ratio of cytosine and guanine is a 1. Similarly, adenine and guanine equal to thymine and cytosine. Adenine plus cytosine is equal to guanine and thymine. So these are the rules given by Chargaff. Now we have to understand double helical structure of DNA molecule. So in DNA molecule, double helical structure of DNA molecule, first we have to understand DNA molecule is having the 2 polynucleotide chain. DNA molecule is having 2 polynucleotide chain. So this type of one chain, this is DNA is the delicate molecule, so we are giving support at center axis. So the DNA is having the 2 polynucleotide chain. Polynucleotide chain is chain which is formed by the many nucleotide. So one chain is this one, and another chain is the this like of structure. So polynucleotide chain, these are 2 polynucleotide chain of the DNA. One nucleotide chain is this. So DNA is made up of 2 polynucleotide chain, number 1. Number 2, the chain run anti-parallel to each other. This axis is 5th end, so the axis is 3rd end. Here it is the 3rd end and it is the 5th end. It means there are 2 polynucleotide chain and they run anti-parallel to each other, and their polarity is 5 to 3rd as well as 3 to 5th. And the both chain are linked with the weak hydrogen bond. So first point, 2 polynucleotide chain, they run anti-parallel to each other and they have polarity 5 to 3 and 3 to 5. And third, they link with me 2 polynucleotide chain are connected with the hydrogen bond. Second, here backbone made up by, backbone of DNA made up of NB, it means nitrogenous base, pentose sugar and phosphate molecule. So if you will see this backbone, DNA is made up of what? The internal structure of DNA are nitrogenous base. This is nitrogenous base, adenine, thymine, cytosine, guanine, guanine, cytosine, thymine combining adenine, adenine combining with thymine. So what is this? Backbone made by this nitrogenous base. This is the nitrogenous base, these are also nitrogenous base. Pentose sugar, I'm indicating with the S, adenine S, sugar, pentose sugar, and phosphate molecule. It means this phosphate, and this phosphate molecule. Here also phosphate, and phosphate molecule. It means if I am indicating here adenine double bond thymine, then here is a sugar, here is also sugar. Then second C combining with the G, sugar, here is also sugar, and this combining with the P. So this type of endoskeleton structure, here adenine plus sugar, it is nucleoside, then added with the phosphate, it is one nucleotide. So this is also another nucleotide, this is another nucleotide. So it is, there are many nucleotides, so it is called polynucleotide. So backbone of the DNA molecule is made up of what? Nitrogenous base, pentose sugar, and phosphate molecule. In this DNA molecule, adenine always combining with thymine with the double hydrogen bond. Adenine always combining with thymine. So always you remember, adenine combining with thymine means thymine combined with adenine, or adenine combined with thymine. So adenine combined with thymine with the double hydrogen bond, whereas cytosine combined with guanine with the triple hydrogen bond. So this is the basic structure of the double helical structure of DNA. A combined with T and C combined with G with the triple hydrogen bond. Now, these two molecules, two polynucleotide chain of the DNA, they coiled each other. And when they coil, they form the helix. Here I am mentioning, this one is the one single helix. Here, in each helix diameter 3.4 nanometer. So this is the one helix structure and 3.4 nanometer. In angstrom, if you will write 34 in angstrom. So first one, what is the diameter of the helix of the DNA? Helix of the DNA, we are talking double right-handed DNA molecule. So helix diameter is 3.4 nanometer or 34 in angstrom. And each helix is having the 10 nucleotide. 1, 2, 3, 4 and the 5. 1, 2, 3, 4 and 5. So each helix there are up to this one 1, 2, 3, 4, 1, 2, 3, 4, 5, 2, 3, 4. Each helix contains 10 nucleotides. So the distance between each nucleotide, between two nucleotides will be 3.4. It is divided by 3.4 divided by 10. So it will come 0.34 nanometer, or we are writing 3.4 in angstrom. Okay. So first one, the double helix structure, each helix diameter is 3.4 nanometer and each helix there are 10 nucleotides. What is their nucleotide? Here this first, second, third, fourth. Okay. So Distance between these two nucleotides is the 0.34 nanometer, and these two structures are the coiled structure. So overall, if you will see in this diagram also, in this picture also, you can see the DNA molecule is having the two polynucleotide chain, and these two polynucleotide chains are coiled to each other, and they are anti-parallel to each other. The polarity 5 to 3 and 3 to 5. Then basic structure of the DNA. Basic structure of the DNA is made up of nitrogenous bases, pentose sugar and phosphate molecule. Adenine combined with the thymine by the double hydrogen bond, and cytosine combined with guanine with the triple hydrogen bond. Each helix structure diameter is 3.4 in nanometer, and each helix, right-handed DNA we are talking, there are 10 nucleotide, and distance between nucleotide is 0.34 nanometer. Similarly, the diameter within these two strands, it is 5 to 3rd, and this is 20 angstrom. Okay, so in angstrom, if you are referring, here it will come 34 angstrom. Distance between two nucleotides 3.4 angstrom, and this between two strands it is 20 angstrom. So it is all about the structure of double helical structure of the DNA molecule proposed by the Watson and